Hello everyone, welcome to a special episode of Katie the Science Lady. I'm Mrs. Jacobson and today's topic is practice with protein synthesis. So let's learn together. All right, today we're going to get a little bit of practice in with the different parts of protein synthesis. We're going to take the gene for hemoglobin, which is the protein that helps carry oxygen through your body in your blood cells, and we're going to turn that into some mRNA and then into our protein. And I'm going to show you this process. First things first, we have to start with our DNA. It's in the nucleus, and in order to get our DNA message out to our ribosomes in the cytoplasm, we have to make a messenger called messenger RNA. So mRNA, we are going to use our base pairing rules, but they're slightly different. T pairs with A, but A now pairs with U. There are no more thymines in mRNA, so when we do these, we need to make sure that anytime we want to put down a T for mRNA, we actually write a U. C and G still pair with each other. So let's go ahead and do these base pairs. T pairs with A pairs with U, C pairs with G. CAC becomes G, A pairs with U, C pairs with G. CAC, CUG, T pairs with A, G pairs with C, A pairs with U. GGA becomes CCU. CTC becomes GAG. Again, same thing here, GAG. TTC becomes AAG. AGA becomes UCU. CGG becomes GCC. CAA becomes GUU. And then our last one here, TGA becomes ACU. After that, there's a stop codon, so we can put stop again if we want. So now we've made some mRNA. The next thing we need to do is use a wheel for our codons, or a codon chart to figure out the amino acid sequence that we're creating here. I'm going to go ahead and write these out, but you could feel free to pause right now and look these up on your own if you want to. AUG is our start codon. After that, we have our amino acids for valine, then histidine, then leucine, then we have threonine and proline. Then we have two in a row that are the same, and they're both glutamic acid. After that, we have lysine, then serine, alanine, valine, and threonine at the end. So this is our normal segment. This is what we're looking at. However, sometimes there's a mutation, and it's called a point mutation, and it only changes one letter. But I'm going to show you how it turns our normal red blood cells from these nice looking discs into a flattened sickled cell that makes it really difficult to travel through the body. So sometimes people with sickle cell can have complications with their breathing. They can get blood clots really easily and have other issues because their blood cells aren't shaped the same. Let's take a look at how this changes. In our first sequence right here, CTC, it's going to go ahead and change. So we're going to substitute this. Instead of a T here, we're going to switch it to an A. Instead of CTC, it's CAC. That's going to change our mRNA. Instead of an A down here, we're going to have a U. Now, just by doing that, that changes this entire amino acid. It's not glutamate anymore, glutamic acid, sorry. Now it's going to be valine. One small change of one letter, one point mutation, causes the amino acid to change, and therefore the entire shape of the protein to change.